Hi, how's it going? This is Harley Genter from biology, I guess, 009, Fall, View College. I'm a business major, and the best way that I learn is by speaking to people and just transmitting information so that even masses can understand something. So I decided to teach protein synthesis to you using YouTube, so I can learn better, and hopefully I can teach you something along the way. So here we go. As we all know, in order to understand protein synthesis, we need to start from the very beginning, so from scratch. Let's talk about DNA. So DNA is a double helix model of proteins. So it's two strands together, and they ring together, kind of like this, like a knot, and they wind around each other, and they're made of nucleotides, and these nucleotides are made up of sugars and phosphates, and the phosphates and sugars are kind of like the base of the structure. So they're what holds the DNA together. The structure of the DNA is made up of sugars known as deoxyribose, and also like a phosphate group, which is used to make the energy needed to build DNA. So kind of like an engine, deoxyribose and the sugars drive it, so keep it going to build DNA. DNA is made up of adenine, thymine, guanine, and cytosine, and all of those are nucleotides. They bind together to make a strand of DNA, which, as I mentioned, is like that. Adenine is referred to A and always binds with thymine, which is referred to as T. At the same time, cytosine, or C, always binds with guanine, or G. It makes sense. A to T, G to C. So if I gave a pattern strand as G, C, G, A, T, the other hand would be C, G, C, T, A. So G always goes to C, C always goes to G, A always goes to T, T always goes to A. That's how it goes. So the binds are always the same. One example that I could give is if a complementary sequence was ATGG, the secondary strand would be TACC. So as we know, DNA replicates, and that's why there's so many cells in our body, and it just forms this huge mass, which is us. We are DNA. We're made of DNA. The base pairs that we start with replicate, and pretty much DNA continues to copy and divide. And this process of copying and dividing is known as DNA polymerase. And in addition to polymerase, there are enzymes which copy strands and fill in the gaps. So DNA and RNA are different. For instance, DNA is used as a model to make proteins. This process involves making proteins, and that's called protein synthesis. So that's what we're here for, protein synthesis. And to begin the protein synthesis model, we start with an enzyme called TRNa. And what it does is it connects itself to other amino acids that are adjacent to other tRNA molecules using ATP, which is its energy source, so to speak. After that, a small ribosomal subentity attaches itself to the 5 ends of the mRNA strands. Okay? So it attaches itself to the last 5. After this process, it moves along the mRNA until it starts a codon. This is where the first tRNA and the largest of the ribosomal combine. Next, the tRNA and anticodonal fall into place with the other ribosomes, okay? And the first of the tRNA drops into amino acids and breaks off, then leaves to pick up other amino acids. Finally, the tRNA drops off their amino acids and form the chain of amino acids linked by peptide bonds. And last but not least, the ribosome reaches a stop codon and releases the finished polypeptide. Okay, so I hope I taught you a little bit about beginning DNA, RNA, and the process in which protein synthesis leads to the replication, copying of protein, and finally DNA. In the same way, if you can imagine, this process is continued millions and millions of times, and it's said that for every 10 million different times that this occurs, there are times where there are mutations. Mutations are pretty much like a mistake, and if you can imagine, with so many different divisions and copying, it would be impossible for there to not be some kind of mistake. These mutations lead to maybe even if it's just 1 out of 10 million, these mutations can be caused by cigarette smoke, eating paint, something crazy like that, even just by mistake. And those can lead to different diseases and negative outputs. And of course, these mutations don't happen very often, but when they do, it can be bad. So finally, I've taught you about DNA, I've taught you about ribosomes, tRNA, mutations, and all of the different components of protein synthesis. And I hope I've taught you something, because I've definitely learned something from this activity. And yeah, this is Harley Chester signing off, and yeah, peace.